Welcome back from lunch, everyone. I hope that all the wonderful meat they're feeding us is not going to cause you to be asleep. Hey, it is really a pleasure and honor to introduce our keynote, Elaine Fuchs. I am so glad she agreed to talk with us here and present us some of her work. Elaine is a true trailblazer and legend. She's really one of the founders of stem cells, and basically, all the biology that we're studying here today, really leading the understanding of skin as a tissue, and everything that you need to do, all the questions that we're trying to query about of how does the tissue work, how do the cells work together to function as a tissue, to maintain a tissue in homeostasis, under repair and injury, and in disease. Elaine has really led all of them. She's a quick adopter to all the technologies to try and address her questions that are very deep in biological, jumping on single cells when they first came out. And of course, there's nothing more important in single cell than imaging and microscopy. And Elaine has some of the best, as they call it in my lab, eye candy, beautiful microscopes with beautiful technical expertise. Now, all this comes with a lot of accolades, and I really can't tell you all of them because that'll take up Elaine's entire hour. She started as faculty at U Chicago and then moved to be Crawford Endowed Professor at Rockefeller. And also, she's in the Howard Hughes Medical Institute, I believe, for about 35 years. That means she survived the rigorous renewal process time and time again. She got, as I said, too many awards for me to list. I will mention the Medal of Science, which is as good as it gets. Also the Pezkler and Clowers, which is the two biggest prizes for cancer research. She's inducted into the National Academy of Sciences, National Academy of Medicine, and just to keep it international, the Royal Society, and I'm just giving you a small sample, about a fourth, a fifth of her prizes and accolades, but I think her biggest achievement is over 100 amazing trainees, postdocs, and graduate students that have moved on to become rock stars and admirable leaders of science in themselves. I think very few can boast a lineage like Elaine. And hopefully in her keynote, you'll be able to see a little bit of what makes her so great. I'm so pleased that she will be talking to us. Elaine, all yours.
Okay, can everybody hear me? Hopefully. Just give me a shout, one shout, and I'll make sure that I just want to make sure everybody can hear me. Maybe Elliot, can you just let me know?
We can hear you loud and clear.
Okay, good. All right, so let me just begin by thanking Donna for a very kind introduction, and really all the organizers for inviting me to this incredible meeting. I really wish that I could get myself up at 2 o'clock in the morning to be able to participate. What I really wish is that I was there in person. Next time, hopefully. And today I'd like to tell you a little bit about how our work over the years has really interfaced with high throughput technologies, and how we can use this information as really a ping pong game to be able to dig deeper into understanding the biology, mostly of barrier epithelial stem cells and their niches. And so that's what I'd like to tell you about today, and hopefully I will be able to do that. I'm just gonna do this, and I hope this goes back. We've been practicing this, good. So if we start with the power of regeneration, it is huge. In a single day, we're losing 50 to 70 billion too, but billion cells, and those have to be replaced. And it's really our stem cells, our tissue stem cells, that are able to do this job. And the barrier epithelial stem cells do it better than anybody else. And in fact, it's because those tissues, our intestine, our skin, esophagus, lungs, they're all at the interface between our body and the external environment, and they maintain niches of resident stem cells to rejuvenate their tissues, to confront pathogens, to repair wounds. And in the skin, We know that there are epidermal stem cells. They're located in the innermost layer of the epidermis at the interface with the mesenchyme, and they periodically commit to differentiate. They produce transit amplifying short-lived progeny that then move upward and are essentially sloughed from the surface of our skin, and that's going on continually. In addition, there's another compartment. There are actually several compartments of stem cells that exist within the skin, and one in particular are the hair follicle stem cells. They're these blue ones in a niche that is called the bulge. And these stem cells, for all practical purposes, spend most of their time in quiescence, but in a cyclical fashion, they can be activated to regrow the hair and essentially regrow the hair follicle and the hair. And then if we look at the crypt in the intestine, we have another compartment of stem cells shown in green here. And these stem cells are, again, continuously active, in this case, producing a monolayer of different kinds of intestinal epithelial cells that are each dedicated to particular jobs. And so epithelial surfaces set high demands for stem cells. And the stem cells differ in different tissues and within a tissue in distinct local microenvironments called niches. And it's really the crosstalk that exists between their neighboring cells and the stem cells that define their behavior. And it's really a two-way street. This is going on constantly as an exchange between different populations of cells within the niche. So epithelial stem cells of the skin have a common embryonic origin, and yet they're defined in their task and their gene expression by their niches. And the hair follicles have really been the ideal system to interrogate the transition from stem cell quiescence to tissue regeneration. The stem cells spend much of their time in this state, with the stem cells shown in purple. There are primed stem cells in this structure called the hair germ at the base of the bulge. They are interacting with a specialized mesenchymal component called the dermal papillae. And it's this exchange that occurs over sometimes months on end that eventually builds up the key factors that are necessary to drive the activation of the stem cells. Once the stem cells are activated, they produce these short-lived progeny that continue their crosstalk with the dermal papillae as the hair follicle grows downward and eventually forms the 7 concentric layers of the hair follicle, the hair, and its channel. And then eventually this undergoes an exhaustion of the short-lived progeny. There's a destructive phase, bringing us back to quiescence again. And as I said, the quiescence can last initially for several weeks in a mouse, then for several months, and then for multiple months afterwards as the quiescent period gets longer and longer as we age. And so as high throughput technology came on the scene, and particularly when single cell analysis and ChIP-seq analysis came on the scene, we began to adapt that technology to be able to study in vivo how the stem cells undergo these kinds of transitions. And again, the hair follicle was perfect for this examination. In single cell RNA-seq that we did, we identified a number of different transcription factors that are specifically active in the quiescent stage, along with a number of different classical stem cell markers that you see here. And what we also learned is that all lineage and differentiation pathways are off at this stage. And when we look at signaling pathways, we find that BMP signaling is high. And if we ablate BMP receptor activity, we mobilize the hair follicle stem cells, and that Wnt signaling is low. And here, if we ablate beta-catenin, the stem cells sit there in their niche, but they never do anything. They're still viable. And then if we ablate TCF3 and 4, the 2 transcription factors expressed in the quiescent stage that are necessary to interact with beta-catenin to make a change in state, we find that when we ablate those, we precociously activate the hair follicle stem cells, suggesting that the role of TCF3 and 4 in this particular context is to raise the bar of activation of Wnt signaling. And I'll get back to that a little bit later. So now what happens when we go from a quiescent state to an activated state at the beginning of a hair cycle? And again, we can purify by fluorescence activated cell sorting all of these different populations and then apply single cell RNA-seq analysis that really drills down into the molecular biology of these cells. Again, we find that BMP signaling in this case goes from high to low. BMP signaling is downregulated for activation of the stem cells, and Wnt signaling goes from low to high. And the switch in BMP and Wnt at the beginning of the switch is really essential for driving that switch. And we're still uncovering new aspects about that. We also see that the suppression of those transcription factors that I described to you in the quiescent state are now downregulated, and we see new transcription factors that are now activated. Two of them are interesting, LEF1 and TCF1, because those two are interacting partners for beta-catenin. And so it's curious that you get these switches. In this case, LEF and TCF1 interact with beta-catenin in order to activate or fuel the driving of the transit amplifying cells and ultimately their differentiation into the 7 concentric rings of the hair follicle. So at the heart of this is really this interaction between the specialized mesenchyme and the stem cells. So what kind of lessons can we learn from looking at different epithelial stem cell niches? Let's look at the intestine. And in the intestine, stem cells use the same signaling pathways, BMPs and Wnt's, but now they're in reverse. Wnt signaling is elevated in the crypt, BMP signaling is low in the crypt. Concomitantly, opposite to the quiescent stem cell niche, and actually more similar to the primed hair follicle stem cells at the bulge base, these cells are proliferative rather than quiescent. And context really matters, because when you really compare the intestinal stem cells and the hair follicle stem cells, even in their prime state, there's still fundamental differences between these two stem cell types. What does that tell us? It tells us that we really need to know more about the impact of the environment and the niche and the chromatin dynamics involved in order to understand how these different choices and these gene expression differences are rooted. And so when we now applied ChIP-seq, and in the early days, it was billions of actually millions of hair follicle stem cells that we had to purify and characterize, which was the major limiting factor, because we wanted to look at everything in vivo. We needed to understand how stem cells behave in a niche. And so when we did that and looked at the transcription factors and where they bind, it's interesting because they all bind in these short stretches of DNA that are located within these larger open chromatin domains. We called them at the time epicenters, but these are regulatory regions, as I'll show you. They also bind various different, or are modified by various different histones that are binding in that region, and also mediator, which brings the enhancer and the promoter together. I point out that all the sequence motifs are there as well. So it's not just a matter of a complex binding to one sequence motif, they're all there. So they're all in these concentrated regions of chromatin. And when you look across a larger super enhancer, you see that there are many of these. Honing in on them, there's additional interesting aspects, and that is that it's not just the transcription factors that are binding in these regions, but it's also the DNA effectors of the key microenvironmental pathways that are binding. Phosphosmad-1 for activation of BMP signaling, and remember we're dealing with the quiescent stem cell niche, and then TCF3 and 4 that are raising that threshold for Wnt signaling in the quiescent stem cell niche, and they're binding as well. So what that makes these domains are molecular zip codes for who I am and where am I. I'm a quiescent hair follicle stem cell in a quiescent niche. So it's only about 350 genes, about 5% of all the expressed genes by hair follicle stem cells that are regulated in this way. And yet they're the key genes. If we look at the genes, they're all the transcription factors that are expressed in the quiescent state. There are also the BMP and the BMP receptors, the Wnt inhibitors and Wnt receptors, and so on and so forth. So these are critical genes that are being regulated by this very interesting regulatory mechanism. And so what happens then when we put these stem cells into culture? Stem cells have to be able to be attuned to their environment and adjust, and that they do. And when you take these stem cells and place them into culture, what we found is that even though these stem cells can be passaged endlessly, they still can make hair when challenged, as well as the other cell types afforded to it, the sebaceous glands and the epidermal stem cells. But in addition, when we look at those super enhancers, we find that they're dramatically changed. Again, 95% of the genes are still there, they're still hair follicle stem cells, but these regulatory regions now are changed. And when we look at the differences in those epicenters, we find that in fact, those differences are that the quiescent stem cell transcription factors are not there. But now we find that transcription factors that tend to be activated in a wound-like state are there. And indeed, when we put cells into culture, they're in high serum-rich, growth factor-rich media. And then we went to the wound state in vivo and showed that many of these transcription factors and super enhancer programs are activated in a wound state when the stem cells have to leave their niche and migrate upward to the wound bed. And so we can demonstrate the physiological relevance of these chromatin landscapes by virtue of cutting out these epicenters from the super enhancers and using them to drive EGFP in mice. And we've developed over the years a very rapid way of making transgenic mice for the skin, where we simply inject the amniotic sac of an early stage embryo with lentivirus, and it's directly taken up by the unspecified epithelial progenitors and thereafter propagated stably. And so in a matter of days, we can do what used to take us close to a year to do. So if we do that, we can see that the epicenter from a quiescent hair follicle stem cell niche is only active in a hair follicle quiescent stem cell niche. And if we correspondingly take one of those in vitro epicenters, we find that it's not active at all in the unwounded state, but if we wound the skin, now it's activated. And so these data give us functionality associated with simply the high throughput analysis. And so we've also looked into malignancy. Similar principles apply in malignancy as stem cells sense alterations in their tumor microenvironment. They're interacting. An oncogenic stem cell interacts with its environment, and that communication dialogue leads to tumorigenesis. We also see the same principles that I've already discussed to you applied there. There's an added twist when we deal with inflammation. And when we started to look at this more carefully, we began to realize that in response to inflammation, if you get lots of different changes, but if you let the phenotype subside, if you let the pathology subside, go back to normal, no more inflammation, and you look, there's still lingering regulatory elements that were opened up months before that maintain an open state. And another twist was that we began to realize that stem cells utilize these memory domains to keep a catalog of their past experiences, and they rapidly respond the next time when they encounter a similar experience. Stem cells remember when they've left one niche and moved to a new one. They remember when they migrate into a wound bed. They remember when they encounter inflammation. And that memory is embedded in the chromatin. And the only way we could have realized this is using a combination of high throughput approaches and biological functional approaches. And so we've really been working in this area for quite a few years now and are very intensively working on this, because some of these chromatin peaks, they start with 40,000 peaks opened up in the peak of inflammation. But if you let inflammation reside, you see that these peaks in red are still there 6 months later. 6 months later in a mouse's life is basically 5 to 6 years in human life, that we have memories going way back to when we were exposed some years ago, presumably. So we can again show that these are real functional elements that are sitting there in the chromatin by cutting them out and doing the same experiment, showing that in 3 different examples, in the absence of inflammation on the top row, there's no activity. But as soon as you give an inflammatory stimulus, the activity turns on with the transgene. And so we've looked over the last year, and I don't have time to go into it in detail, but we published it last year. What is the mechanism that underlies memory establishment, memory maintenance, and memory recall? And interestingly, when you look at these regulatory elements that are inflammation sensors, the same kinds of principles apply to what I've already described to you. We find that in order to establish memory, STAT3 and FOS, coupled in its heterodimerized state with JUN, are basically essential to open up and establish the memory domains. STAT3, we think, binds directly to nucleosomes, whereas FOS and JUN act as chromatin remodelers to activate transcription at these sites. But then, once the inflammation has subsided, STAT3 and FOS are no longer there. And so what's happening? What's keeping the chromatin open? And there we showed that at that early stage, once you establish and open the chromatin, other stem cell transcription factors come in and bind to the chromatin. There are binding sites for them. And then when the inflammatory transcription factors are no longer there, they still keep the chromatin open. And then the chromatin is open for months on end. What's involved in reactivating the chromatin? And there we showed that all you need for that step appears to be FOS, because and FOS is induced in a whole variety of different stress-induced factors. And so all sorts of different stimuli can activate FOS. STAT3 is no longer required because the chromatin's already open, and that's what STAT3 did. And so you can get rid of STAT3 at that stage, and you find that memory recall still works as long as you've got FOS. And so this begins to explain a whole variety of different human diseases: psoriasis, atopic dermatitis, asthma, inflammatory bowel disease, because all of these diseases are typified by having hyperproliferation at the first assault. Hyperproliferation subsides, everything looks normal. All of a sudden it comes back again. It's not necessarily the same stimulus that activates it the next time around, and it comes back with a vengeance, and the chromatin is already open, so it's very rapid to reopen it again. And so I'd like to now switch and in the latter part of my talk, give you another flavor for the importance of really combining high throughput technology with biology to really understand what's going on. Presumably, with the work that I've already described to you, you now have a strong appreciation for the importance of the microenvironment in operating on stem cells. Stem cells are really responding to recording their experiences and responding to new changes in their microenvironment. And so what's important there is their interactions with their niche. And for a long time, we were just focused on those dermal papillae in the hair follicle as being important, but we began to realize that there's a lot of things going on in a stem cell niche, and they can be very complicated. I'll give you a flavor for some of this in this study. So several years ago, Sherry Cohen in my laboratory had been interested in studying the vasculature. Is the vasculature changed around a stem cell niche to be able to provide nutrients to the stem cells? And so in order to be able to look at 3D interactions between stem cells and their niches, we exploited a new technology at the time, developed in part by my former president, Mark Tessier-Levine, for tissue clearing. And that allowed us to do tissue clearing and 3D whole mount immunofluorescence imaging. And from that, we could get a picture of stem cells, not just in a cross section interacting with various different niche components, but now looking at 3D. And at 3D, you start to see beautiful interactions between stem cells and their niches. And those niches now compose three-dimensional networks of things like neurons and blood vessels and lymphatic vessels. But it turns out that in the hair follicle, the lymphatic capillaries, the draining part of the vasculature, turns out to play a very integral role. And when we first got a glimpse of this, which was off everybody's screen, radar screen, including our own at the time, we began to realize what was going on. And so here in red, you see lymphatic capillaries. And this is looking now at down at a cross section, at a thick section, through a thick section of the hair of the skin. The green are the stem cell niches of the hair follicle. And you can see that every stem cell niche has associated with it a lymphatic capillary network. And what that does is basically integrate the entire set of stem cell niches across the tissue. And when you think about tissue behavior, that's exactly what you want. You want to have all the niches integrated, doing the same thing to be able to perform a particular function, whether it's a wound or in this case, mouse hair cycling. And so in 3D, you can really start to see this very clearly then. Again, the lymphatics are in red and the stem cell niches are in green. Another curiosity that you might notice here, which we don't understand yet, is that the lymphatics are on one side of the niche, not both sides of the niche. And so that's something that we can talk about, but we're currently trying to understand at a molecular level. Another really curiosity about the system is that when we followed it through the hair cycle, we realized that lymphatic capillaries are not always connected to the hair follicle stem cell niche. And at the start of every hair cycle, the lymphatic capillaries detach. So here they're attached, starts the hair cycle, they detach, and then they readily come back and attach again. And this is happening over here, the reattachment during the active stage of the hair cycle. So during anagen. So it's not simply that there's a physical force that breaks the lymphatics away, but here is a controlled interaction. And that control we learned was at the level of the stem cells. The stem cells are coordinating that action by virtue of producing one factor called angiopoietin-like 7 during the quiescent phase of the hair cycle, both at the quiescent phase, the resting stage, but also in these later stages when the stem cells have gone back to quiescence, but the transit amplifying cells are fueling the hair cycle. And so angiopoietin-like 7 is there, and we showed by knockouts that it's important with regards to keeping the stem cells attached with their lymphatic capillaries. On the back end of that, at this stage, when they detach, then they're expressing the stem cells when they get activated. Remember, gene expression changes. Now they express angiopoietin-like 4. And again, we showed by knockout technology that angiopoietin-like 4 plays an integral role in this transient, short, brief period of detachment. And so that's a curiosity to keep in mind, that it's only during this short window where we see detachment. That short window is precisely the time at which the hair follicle stem cells themselves are activated to proliferate, to self-renew. So once the transit amplifying cells are produced, they fuel the hair cycle and the stem cells go back to quiescence. But just before they do, they undergo self-renewal. And it's during that period of time that we see this detachment. So if we detach permanently the lymphatics using conditional knockout technology, these quiescent stem cells shown in green here suddenly now produce new hair follicles, and they undergo activation from quiescence to activated state and fueling the hair cycle. So they're functionally important in this, as we showed, angiopoietin-like 7 and angiopoietin-like 4. So now we've got a lot of questions. How do lymphatics govern the stem cell niche? It could be by controlling BMP or Wnt signaling, perhaps controlling the levels of signaling that are produced by lymphatic endothelial cells. That's a possibility. And it could be that it's immune cell trafficking, because the lymphatics are draining, but they also transport resident immune cells to the lymph nodes. And so that's a possibility. It could be that the stem cells are in some privileged immune site, or that they're being controlled, the switch is being controlled by immune cell trafficking. It could be that it's pressure, that it's fluid pressure. The lymphatics are important in drainage, and you know that in lymphedema, which is after surgery, if you've got this swelling of tissues, that's because the lymph nodes were cut and they have to be repaired. And so it could be that it's simply fluid pressure that's controlling the switch in angiogenesis, an interesting thought that we haven't really yet delved into in detail. And it could be that the lymphatics are simply protecting the stem cells, that we know that, and as we look through the hair cycle, that there's actually a brief window of increased DNA damage, and that's the stage at which the stem cells are proliferating and at which the lymphatics are detaching. And so that's another possibility, many possibilities, and we still have a lot to do with regards to digging through the biology. But another way in which we can dig through the biology is to look at parallels, again, to additional systems, and in this case, looking at the intestinal epithelium. And herein is one of my favorite collaborations of all time with Donna Pear's laboratory, Qingyi Xu in Donna's laboratory, and then three postdocs, two postdocs and one graduate student, Marina, in my laboratory. And here, the first thing that we wanted to do is to really look at, is there a parallel? And people at the time had focused on lymphatics, but they've been focusing on this yellow structure here called the lacteal, which in the small intestine participates in lipid absorption. And what we see is actually there's a lymphatic network down here that has largely been ignored over the years. But curiously, lymphatic dysfunction occurs in inflammatory bowel disease. And like the bulge, hair follicle lymphatics, the crypt lymphatics had been largely ignored. And so the first thing that we did was to use our 3D imaging and tissue clearing to take a look at the lymphatic structure. And indeed, what we see is at the base, this is now looking front on the bottom of the crypt niche. And what we see is this intimacy between the lymphatic capillaries in white and the stem cells of the intestine in green. And at the ultrastructural level, again, you can see that there's this really close association that has been largely not paid attention to that exists down here at the crypt. And so we then wanted to do these same experiments. What happens when we lose lymphatics in the intestine? And when we do that, what we find is that after acute lymphatic ablation, that we find stem cell mobilization. We've monitored by pulse chase the flux of stem cells from the stem cells to the transit amplifying cells and upward to the top of the villus. And we showed that all of this is accelerated in response to ablation of the lymphatics. Interestingly, something that doesn't occur in the hair follicle stem cells is intestinal stem cell exhaustion. We see that not only is there proliferation, but eventually exhaustion of the intestinal stem cells. We're particularly intrigued by this because in the hair follicle stem cell niche, there's a buffer, there's a quiescent buffer that is operating on the stem cells to dampen down proliferation. That's missing in the intestinal stem cell niche. And what we find is that if we challenge the hair follicles and just pluck the hair constantly, and the stem cells have to work constantly in a way they don't want to, we see stem cell exhaustion. So we think that there's an importance of the niche in terms of controlling stem cell exhaustion. One of the ways that is operating is by the lymphatics, but the other way to maintain stemness, but the other way is by virtue of having this bulge niche, and muscle stem cells and and hematopoietic stem cells are also undergoing these quiescent states. And so the next question that we could address that we couldn't address with the hair follicle stem cells is this a direct effect? And with organoid culture for the intestine, it was very easy to do so. We could show that the stem cells exposed to lymphatic endothelial cells have increased organoid forming efficiency. They have decreased differentiation and maturation. So here it's very clear, both in the hair follicle and in the intestine, that the role of lymphatics in part is basically to put the brakes on differentiation and to maintain stemness. So conditioned media also works, which says that there's secreting factors that are functional in this. And so how do we find them? And so here is where we really teamed up to look at spatial transcriptomics, because single cell RNA sequencing gives us depth of coverage, but spatial cartography is necessary in order to dissect the complexities of a stem cell niche. And of course, one of the problems that everybody faces in the field is that 10x Visium spatial transcriptomics has spots, barcoded spots that are 55 microns in diameter, which often, depending upon where the cells are over that spot, can include up to 10 cells. And so you get bits and pieces of gene expression profiles, and you really need a way to deconvolve all of that. And so what we did on our part was to use fixation for the tissue. Here you see the intestinal tissue on one of those grids. We stained with our various different markers, including the stem cells in green. And you can see then on these grids where the intestinal epithelial is, where the villus is at the top and where the stem cells are in the bottom. And so what Tiny did was to integrate single cell RNA sequencing with spatial transcriptomics to reconstruct the cellular composition of each spot with higher resolution. He did that using a Bayesian statistical model that uses single cell RNA-seq from the intestine to infer the cell types within each spatial transcriptomic spot. So we did single cell first, and then using Bayesian prism, where we could really identify what cells are within each spot. We could get this information, how it differs spatially from the top of the villus to the bottom where the stem cells are, and below where the more stroma is. And so the next thing that was important is to project all these spots onto a common axis. And what that does for us is you take hundreds of spots like this, and you deconvolve them, you know what cell types are there. But now, if you project them on a common axis, you can now look at gene expression patterns from the top to the bottom, and you know what cells are there. And this really turned out to be powerful. Spacefold, which Donna can talk to you about, is just really phenomenal in what it can do. It can tell you not only where the cells are, but also what gene profiles they have. And the gene profiles can differ when you're looking at a lymphatic cell in the lacteal versus a lymphatic cell in the intestinal crypt. And so we know that really it's a two-way crosstalk between stem cells and their interacting partners. And so here is what it looks like in beautiful full regalia. Each one of these dots is actually a cell of a particular type, for instance, the lymphatic. And you can see the lymphatic endothelial cells at the crypt base, and you can see the lymphatic endothelial cells in the lacteal. And for each of these, you can see where spatially these different cells are located. But now, if you look at this from the lens of gene expression, you can now see where gene expression is occurring. So I just show you for LGR5, this spatial cartography shows you the villus on the right and the bottom of the tissue on the left. And here is where the stem cell crypt niche is, and that's where the peak of LGR5 expression is. And now if you take a look at what's going on in the lymphatic endothelial cells, for instance, that are surrounding the crypt niche, what we see is really amazing insight. So here are the relative expression patterns that you see. CCL21A is a well-known lymphatic endothelial cell gene. Here are some others that weren't well-known. R-spondin 3 was always thought to be expressed by the fibroblasts surrounding the intestinal stem cell niche, but in fact, even though it is, it's not nearly expressed as highly as is the lymphatic endothelial cells. And then you look at others like this one, relin, which is only expressed in lymphatic endothelial cells and not expressed anywhere else. And when you look spatially what you find is that the classical control, CCL21A, is located in the lymphatic endothelial cells at the crypt niche. We see relin is located there, and we see our spondin is located there. So again, you start to get spatial information about gene expression patterns. And the only way we could get this information is by combining single cell RNA-seq with spatial transcriptomics and applying spacefold. And then, of course, you have to do biology to tell you, is this actually functionally important? And so I'll just give you a little vignette here with regards to relin. First of all, is it really expressed or concentrated at the crypt base? This shows you immunofluorescence with intensity maximums, and you can see that that is highest in the crypt base relative to the lacteals. And if you then take a look at three-dimensional imaging, relin is in yellow here, and the lymphatics are, the crypt lymphatics are in red. And that's what we're looking at the very bottom, where the stem cell niches are in blue, which I passed over. We can do conditional knockout technology and look at what is the functional significance of relin. And again, we get very similar results to what we see with lymphatic ablation. And that is, there's proliferation that's accelerated. The proliferation is accelerated, and their exit, this is in a pulse chase, their exit from the niche is much greater than it is in the control. And if we then look at direct signaling, it is direct signaling because now we can replace the lymphatic endothelial cells in our co-culture with simply relin and show that we can get the same effects on controlling stemness and suppressing differentiation by relin. And so finally, if we look at, let's go back to the hair follicle stem cell niche and look at these gene expression patterns, we find that relin is expressed by both lymphatics and hair follicle stem cells. And remember that hair follicle stem cells and intestinal epithelial stem cells utilize the lymphatic endothelial cells for controlling, maintaining stemness and for repressing differentiation. So we have a commonality with a factor that does that, and with these two stem cell niches and their parallels. Our spondin 3, however, is the major enhancer of Wnt signaling. And remember that in the hair follicle stem cell niche, Wnt signaling is down. In the lymphatic and in the crypt niche, Wnt signaling is high. And when you look at expression patterns of our spondin, they're much higher in the lymphatic endothelial cells than they are in the hair follicle lymphatic endothelial cells. And so, so these data are starting to give us new insights into understanding, in this case, just one component of what is a very complicated stem cell niche. So I can't remember when I started, but I suspect that I'm reaching my time, if not over time. But just to recapitulate this latter aspect of it, because it might be of most interest to this audience, that the spacefold cartography really captures the cellular and molecular landscape of a stem cell niche, a stem cell and the stem cell niche. And it does so in context of the tissue. It can lead to discovery of new signals as you learn from relin, and new niche components as you learn from lymphatic endothelial cells that are not possible with conventional approaches. And despite very different tissue demands, intestinal and hair follicle stem cells rely upon lymphatics to maintain their stemness and restrict tissue regeneration, and relin endows the lymphatics with this signal. And as I described, our spondin is differentially expressed. We don't know why, but we assume that just like the niche and the stem cells, the niche components are also experiencing a different microenvironment, and that must be influencing the gene expression patterns that we're seeing here. So I think at this point, I'll just stop and say that, that effectively the approaches in the future that we're going to in terms of understanding complicated interactions that exist of cells within a tissue is really going to rely upon lots of collaboration between those fantastic computational biologists that really understand the importance of how to drill in to the high throughput data that we and others are generating, and to provide new predictions and insights in doing so, along with those biologists and geneticists such as myself who just learn to communicate with each other. And I think in our collaboration with Donna, we've really both learned how to do that. So I'll stop there and just thank you for inviting me today.
Okay, so questions for Elaine? Do we want to start with, is there anyone online asking a question? No, not yet. Aviv.
One of them, please. Hey, Elaine, first of all, thank you so much for joining us. This was a spectacular talk. I was wondering how you think about more than pairwise interactions in the stem cell niche. So several years ago, we actually did it in ISCs, and I know you have done it for the hair follicle. We found interactions with, say, T cells in the niche that is important to drive this based on environmental signals that the T cells can sense or through cytokine signaling. Now you're showing us beautiful evidence and how the lymphatics are actually providing another kind of signal. How do you see the circuit between the different cells, more than the pairwise interactions, and where do you see the future for that?
Yeah, I mean, it's a great question, Aviv, and what we've looked at is just a two-way street so far. And yet when we look at the receptors for instance, for relin, I mean, beta-catenin is one of those, beta-1 integrin is one of those receptors, but there are two additional receptors. And those receptors are also present on different cellular components. What we can show, which I think is one of the important aspects of dissecting the importance of these different interactions, where you've got one ligand, but you've got several potential cells that could be interacting with that, is the importance of doing these in vitro assays, where we've really drilled down on saying, does relin have an effect on the intestinal stem cells? And that you can do in culture. It's much more difficult to do in vivo, although it's not impossible, because conditional knockouts can really still help us and guide us. We can do conditional knockouts in the epithelial stem cells very easily by our lentiviral mediated approaches. It's more challenging to do in looking at the various different niche components. So that said, there are increasingly better tools for targeting specifically, and I think that will help us. We just lost signal.
We didn't. We see you really well, and we hear you.
Okay, great.
Bruce?
Got it back.
Boy, I think I have a very similar question. This is Bruce Arno. Awesome talk. I have a very similar question, I think, to Aviv. Why in these cycling systems do we not see more autoimmunity generated? And you can see, there's been, I think, a big instance of alopecia areata universalis in post-COVID, but the idea that the immune system is juxtaposed, immune system conducting elements are juxtaposed to these cycling states. Do you see in those autoimmune type conditions any possibility that the system's linked and that there's an abnormality of that structure there?
Yeah. I mean, what's interesting, we've studied some of this quite a bit. We haven't studied alopecia areata, but we have studied we have looked at atopic dermatitis, and we've also looked at the link between structural protein of the skin that's not even expressed until the stem cells have long committed to differentiate, and squamous cell carcinoma. So you'd say, how can these things be linked to one another? And I think what's emerged from our studies and studies of others is that for all of these different, what people always thought traditionally were immune disorders, because they create inflammation and the like. There are two ways to do that. One way is to have an immune-focused problem, and mutations, for instance, in atopic dermatitis have been found in interleukin 13 and interleukin 18. They're yet polymorphisms. They haven't been demonstrated to be crucial in causing the disease, but they're obvious candidates for causing the disease. And then you find a protein, structural protein called filaggrin, which is expressed in the latter stages of epidermal skin development. And what we showed is that you will get the same phenotype, the inflammatory disorder and all, but it's because without filaggrin and this ability of this protein to undergo liquid phase dynamics, if all completely unexpected, if you don't have that, the mutations don't allow liquid phase transitions to occur. What you get are nuclei and organelles left in the outer layers of the skin, and that compromises, it weakens the barrier. And so that leads to the same phenotype of hyperproliferation, but from a very different reason. And for years, people were just ignoring those kinds of mutations, saying it's got to be an immune-related disorder. And I think what we're now realizing is some of the complexities of all of these different inflammatory disorders, where you'd say, why haven't the genetics been figured out by now? And one of the reasons is because there are multiple different ways to create these kinds of conditions. And so now if you add on to that, the fact that immune cells certainly have memory, but the memory that we've discovered in the epithelium exists independently. We can do this experiment on a RAG2 null mouse. We can do the experiment on an IL-17 receptor null mouse. And if we're looking at who retains memory and who activates memory, then what we find is that the immune cells are dispensable. It's not to say that they don't have their own memory, but it's to say they don't have this memory. And this memory causes a rapid response. And so I think what you end up with are these real curiosities. And you think about COVID and say, why is it that adult people, aged people, end up with this kind of a higher rate of reaction? And I look at this from saying, well, if you've got memory and you're harboring a lot of memories of inflammatory experiences, that's what you would predict. And it doesn't have to be any pathogen that you've ever seen before. And we've never seen COVID before. And so I think some of these things really need to be revisited. And with the hair cycle, for sure, you're dealing with a barrier. There's bacteria that can go right down into the orifice. And so, yeah, it's a complicated street. And I can only think that there's a lot more biology to be done.
Okay, Muz?
Muz Hanifa here, thanks for the great talk. I was just wondering, with regards to developing skin, the lymphatics don't develop after a certain stage. And I wondered what you thought about how the stem cells are maintained before that, and also if there was any evidence of stem cell disorders in lymphatic diseases.
Yeah, certainly the stem cells are affected. I mean, in terms of disorders that are associated with lymphatics, and lymphedema can certainly alter the biology of the epidermis. Sherry Gercohen is looking at some of these things now that she's at UCSD. With regards to development, it's interesting where we've been starting to look at that in detail, because the one thing that we know is that in the early stages, you have multipotent progenitors. Actually, they're going to be long-lived progenitors, but the progenitors in the skin start as unspecified progenitors, and the only time at which they start to behave in a particular task is when they form the niche. And the stem cells, again, through crosstalk with their environment, and you can look at it in development, the cells that are moving into the skin are changing all the time during development. Those progenitors the stem cell progenitors only become dedicated stem cells it's right before birth, and that's when they really start to establish a niche. And for the hair follicle, it's even not established until some 3 weeks later, fully established, as immune cells are changing as well. And so I think that these things are all regulated in ways that we still don't understand, certainly for development. We're starting to look at that in earnest, but for the adults, when we look at the complexity of the stem cell niche, we realize that it's not just a matter of local signals that are coming from their neighbors, like the dermal papillae, but now you've got far-range signals like the lymphatics or the neurons that can really respond externally to all sorts of different environments. And so the next level will be how different tissues are able to convey different signals from one stem cell niche in one tissue to another stem cell niche in another tissue, which I think is likely to happen. That's the fourth dimension of this. But again, I think you have to look, it was like Aviv's question, you have to come up with ways where you take the complexity as far as it can go in vivo, but you really need a combination of picking it apart in vitro and going back and testing it in vivo to really understand these things. I don't know if that addressed your question, but it's kind of the gist of it.
Yeah, okay, you came? The final question.
Yeah, Elaine, Joachim Schulze from Bonn, really fascinating. I might have missed it, simple question, the Raylene regulation throughout the hair cycle. Do you know already if that's like the vessels is changing in expression or protein expression function, anything during the hair cycle?
Yeah, I love the question. We've done single cell analysis during the hair cycle, and Sherry Cohen again just left my laboratory within the last couple months, and she's really going to start that up in her laboratory. And I always like to leave my young investigators space to develop their own labs before sort of starting collaborations or doing that. And so we've kind of left those questions for her. And most of the work, the whole work on Relin was really uncovered through our collaboration with Donna and Tingy, because it would not occur if we didn't have that spacefold drilling down to look at these things. And the pairing as well. So I think that it's going to be interesting. We know there are differences from the single cell that we did in the paper that we published, but we really haven't done much with regards to Raylin in the niche. It's curious that beta-catenin, or the beta-1 integrin, I keep getting that confused. Beta-1 integrin is a putative receptor for it, even though there are other receptors. But it's interesting because that's a commonality of virtually every single stem cell niche. So I think as more of these things uncover, people are going to appreciate it, and appreciate the fact that niche cells can also differ because they themselves are in a different microenvironment than they are elsewhere in the tissue.
Okay, just before we thank Elaine, I have to say she's complimented me so many times. Yes, the computation helped, but also the pristine quality of Elaine's experimental data, as well as many critical experimental choices, such as sorting and enriching these critical rare cell types in the niche, were as critical as the computation. And, oh, did she disappear already? I wanted to thank her and give her another round of applause, but...
Thanks very much.
Oh, well, thank you very much. This has been really an incredible day. Just beginning with the series of talks we had at the beginning of the day on different modes of accessing and using data, then continuing, of course, through the first breakout sessions, and of course, obviously that just a really extraordinary talk from Dr. Fuchs. We're actually going to return now to the breakout sessions from before lunch, and this is going to be a relatively quick-hitting report back session. Unfortunately, we really do not have time, I think, to really continue the discussions in depth, but what we hope to do is hear from each of the moderators, the pairs of moderators, on the key points that came out of their session, and then we'll have time for a few questions for each of the, at least one or two questions for each of the pairs of moderators before we move on to the next section. So let's get started then with the first breakout group, which is Chloe Villani and Kirsten Meyer on good practices for making organ-specific atlases. You can use the podium or handheld, whichever.
Maybe I'll borrow. So this is what, okay, cool. Okay, while the slides go up, so we thought it would be a good opportunity to gather members from different bio-networks to try to share good practices, challenges, and opportunities so we could learn from each other. And so, can we just, do you want to start, Kirsten? We're going to try to summarize quickly in 5 minutes. I'm timing ourselves. Go ahead.
Yes, so we came up with a couple of questions that we considered, and of course there was a large amount of discussion, and we're not going to cover everything, we're just going to pick a few things that we thought were relevant. So the first question, we asked whether there are any recommendations or particularly effective way of communicating within bio-networks. And the one key point that really came out is that community building is incredibly important, and this often takes a lead who will put in some very significant time commitment, and in that context it's also really important for that person to have funding, but also then buy-in from the rest of the community to provide the data. At the same time, it's really, while those networks are being established and there's importance to really build that network, the networks need to be open for other people around the community. And then we also wanted to say that the DCP can also really help in terms of exchanging data. And I'll pass over to you.
Yeah, and so I think one of the main challenges was some of the bio-network are struggling to gather data while others are quite ahead, so that community building is quite essential. And some of the other challenges that were related to that beyond the data, so it was all related to data sharing. So some folks do want to share data, but there's different legal constraint, ethical constraint. And so beyond identifying data set, it was, how do we proceed moving forward? So there's actually an ethical working groups who's offering to meet and try to help and standardize and lower these boundaries. The Long Cell Atlas shares some of their good practice, which involved also sharing pre-processing standards. So those that could only share count matrices, could at least process their data exactly the same way. So these are good examples for others to adhere. There is a whole idea of de-identifying the data sets, scrambling the genetic background, which I know the genetic bio-network will not like as a solution, but in some instances, there's no other solutions. It depends on your IRB. And I think that covers it for these main issues. There were many more, but we're trying to summarize the key points.
And then, of course, one of the key questions in terms of building an atlas is cell type annotation, and there are, of course, a huge number of challenges. The first one is that there may or may not be a lot of disagreement on what the different cell types are and what they should be called. But one way of approaching that is, first of all, to have hierarchical annotations so people can agree on a first level of cell type calling and then drill down further. But it's important to state that as yet there's no standardized way of doing this. It can help to really integrate multiple data sets because sometimes this really brings out the biology, but of course there are also instances where batch correcting across multiple data sets also corrects out the biology. So it's a case of trial and error of what really works. And maybe you would like to mention the Jamboree.
Yeah, so within our immune bio-network, we're finding challenge related to our data toward legacy knowledge, agreeing on some annotations. So we are putting together a cell annotation jamboree with the goal of the whole community coming together to analyze exactly the same data set, so we can start having that conversation about cell type annotation. That's open to everyone who wants to participate. It's going to be launched this fall, so stay tuned for this.
And so very briefly, also different networks that have formed then, of course, need to talk together, like, for example, Lung Map and the Human Lung Cell Atlas.
And perhaps to add to this, some cells are shared across tissue. Again, immune cells, for example, are shared across tissue. So we want to make sure that there's consensus in terms of harmonizing the way we approach annotation across tissues over time.
Yeah. And then lastly, we wanted to talk about the gaps that still exist in terms of building the network. The first point of discussion revolved around sampling strategies. So these really need to be worked out organ by organ. And it was also pointed out that the first version of the atlas will really be very, very helpful in terms of figuring out where there are still gaps, both in terms of donors and the types of tissue or cell types like granulocytes that we might still be missing. There was quite a bit of discussion about organ tissue procurement in terms of what's healthy is neighboring normal healthy, but I think we agreed in the end that we will just go ahead, build the first version, and then some of these questions will become more apparent. Another gap is definitely that we haven't really sampled the diversity of the population yet, and I think every network is very aware that that is an issue to address in the future, but again, the version 1 of the atlases will be really well placed to point how that can be extended.
And maybe I'll just summarize the last few things. So most of the modalities being reported are mostly in the RNA space with a bit of protein. So all of us are realizing there's just version 1 where we have to do with what we have and come together and share the best practice for the other modalities. Common coordinating framework, when we asked this question, it was interesting to see how nobody had great answers. So there really is a need to better coordinate around this topic and come up with solutions. And finally, disease. Everyone had different thoughts about it, including disease. But clearly that's a problem to be tackled for version 2. And so sorry to our group if we didn't catch all of your thoughts. We did our best, John, in 5 minutes. Yeah.
And I do actually want to say we did have scribes in all the sessions, so we did actually capture all the thoughts there. And also, I do also want to say that the videos will be available possibly starting tomorrow, so you can actually watch the other groups that you were not in as well. So we have time for perhaps a couple of questions from the audience, including, I should say, the online audience.
And while we ask for a hand up, I thought it was interesting. We asked multiple times our group what were their metrics for success and utility. And it was interesting. I think we asked that question 5 times, and nobody could raise their hand, which I think all of us are still struggling figuring out our data set and bringing them together. And the lack of answers perhaps highlighted that challenge.
I think Donna has a question.
So I wasn't in your session, but I'll give you an answer for the unanswered question. So again, it relates to our session where I think one of the basic questions we need to ask is what type of queries we want, and then the metrics of utilities relate directly to the queries and how accurately and faithfully we're able to execute on each of the queries we want. So the metrics need to be centered around well-defined queries and our ability to answer them with the current available data and computation.
I couldn't agree more, Mary. I think this highlights the need for the computational working group to work more closely, or bionetworks to work closely with the computational working group and vice versa.
I couldn't agree more. I've been jumping up and down and saying that, but I thought that the bionetworks were ignoring me on purpose.
No, we're not ignoring you.
Because I'm so dominant and opinionated.
We love that, Donna. We love that.
I think we'll need to move on.
Yeah? Okay.
So let me call up actually Donna and Nir for the next breakout group, which was on computational querying of the atlas.
So we had a really fun session, in my humble opinion. Really, again, as I said, it really tried to focus around the queries. We had some optimistic stories of really successful queries to the atlas. I think we saw another example of that with Elaine. But really, we sort of brainstormed what queries. We have a list of queries, which I'll show you in a moment. And I have several conclusions. First of all, while a lot of the other working groups were really focused on cells, our queries suddenly pulled us back to genes. And genes were central players in our queries, and that took up much of a discussion. Even simple queries, such as where's my gene or gene program is expressed, as I said yesterday, is complex. We have a lot of work. But I think Aviv was really successful in really pinpointing the problem. We have to really define our queries with pristine accuracy and crispness. And even some of the simple queries, when we got some very talented postdocs and graduate students together and said, okay, define this query in very accurate mathematical and biological language, there's work to do even to define the queries. There was a bunch of queries raised, really good ones. We took a vote to see which are the leading ones, and Nir will describe our favorite queries.
So yeah, so we asked the audience for what would be interesting questions to ask when we have a data set, the Atlas in hand, and we have a list here of most of these questions that came out. So really interesting questions for us to think about it as most of them are open questions and really think that we need to invest in. So the first one was really about just finding marker genes and doing it maybe focused on cells markers to sort cells. Then there was a very interesting question about connecting single-cell genomics to genetics. And
you should focus on our leaders because you have 30 seconds.
Okay, but you just want to finish. Okay. Well, anyways, connecting single-cell genomics to genetics and looking for some interpretation for causal variants and so on. So that's really building in common accessibility from single cells and so on. But our three lead questions here that you can see are, one of them was, how do we use this human cell atlas to contextualize comparative studies? So this is a question that was answered, it was left as a question. Of course, this is something still open, many challenges, but we at least enumerated the two approaches of, can we look at compositional changes, gene expression changes, outlier analysis, and so on. This is really where we are going with that in our thought processes, computational groups. And then there was...
Skip the next one, that was a mistake.
Yeah. And then there was the big, maybe one of the big discussions that we've had was to move from looking at individual genes to actually think about gene programs, right? Gene programs is a collection of genes that are being expressed not necessarily by a single cell, but potentially also by groups of cells that can be of different types. So we first of all talked about how do we even approach this problem? How do we define a gene program mathematically and conceptually? What are the different ways that we can approach to this problem and find these gene programs by matrix factorization, feature selection, so on, all these discussion ensued. How to evaluate how well we're doing in finding gene programs, and in a sense, how do we use it as a baseline for describing what is a cell state beyond cell type, beyond putting discrete labels on cells. So that, in a sense, connects to what Dana said about the gene-centric emphasis that this discussion received. So I'm proud of that. Yeah.
Terrific, thank you. Questions on group 2? Fabian has a question. Yes, Fabian.
Thanks. So the where and the programs totally get that. The last one with the reconstruction, you think is that so tough? I think faithful gene counts, you're just saying it's going to be too average, too not so clear. You think we should be able to reconstruct better, like in terms of more realistic, like, you know, images VAE going to transformers? Is that sort of the thing that was?
Yeah, okay, so maybe I should comment on this last question here. There was this, this was more of a technical kind of question and not a conceptual. It's not a query, if you will, right? And which is, how do we go and think about the data in high dimension, so the gene expression data, the common accessibility data, in a way that is normalized, that is not affected by batch effect and so on. And we had some discussion about variational autoencoders, and this was maybe a bit of a misunderstanding that these VAEs are only able to provide low-dimensional representation of harmonized data, and this is actually something that these, mathematically at least, they are able to provide normalized data in high dimension that we can use to do differential expression and so on. But that's not to say that this is still a very much open area of being able to receive corrected, normalized gene expression data, batch effect corrected, and so on, similarly for common accessibility. And there was a related question on that from CZI about the problems that this generates for scalability. Because if we are trying to impute data and trying to normalize it, we are losing sparsity, and that creates a lot of issues in terms of compute.
Yeah, no, I had a little personal discussion with the CZI gang afterwards, and I'll share with everyone. I think that, again, our way forward, and this is a call to arms for everyone to participate with us. First of all, define the queries very accurately and very concisely in a way that we can turn that into a mathematical and statistical equation. Then, and this will have to be iteratively, because as you get the answers, you realize, no, that's not what I wanted. Then figure out computational algorithms that actually work and get the biology that we really want in a very robust, effective, and valuable way. That will likely be, as the CZI folks complained, computationally inefficient. Then understand the essence of why it works. What is the core reason that, you know, our windabout road that took us somewhere works? Strip all the unnecessary crap off and get an efficient algorithm that CZI can help us scale, or other very effective software engineers.
Great, thank you. If there are no other questions, then maybe we'll move on to group 3. Let me ask Kadi Border and David Asumi Sutherland to come up. And this group was on ontologies, annotations, and metadata.
Yeah, so we had an extremely productive session with lots of really great ideas and input. We couldn't possibly cover in a slide here in the report back. So we've really focused on more concrete steps that we can take to proceed. So one issue we identified was just a general confusion with where the annotation happens and what the data flows are between the various portals that we've got. So we've got CAP, we've got cell by gene, we've got all the integration work that's going on in Fabian Thesis Group in Munich, for example. There's the annotation that goes on in the DCP. So one action for us is to try to come up with some recommended workflows. And we have a sort of example thing here that Fabian suggested as kind of a starting point. In the context of that, you know, so first of all, where do users start by submitting their data and maybe doing some of the annotation themselves? Now we have things like the cell annotation platform. How should data flow between portals? And can we kind of make that a bit more concrete? Should we be encouraging annotation to integrated consensus cross-modal analysis as well as the initial phase of analysis? So a lot of that annotation is coming through in automated fashion. Can we bring the community into the loop there as well? Another important thing we thought was to try and agree on versioning for Atlas data and portals so that when data moves between atlases and annotations potentially move with them, we know we've actually got a record of versions as they go. So I mean, if you were to, you know, work with genomics data, you'd take for granted that you would say, I did my work against this particular reference build of the genome. We need some kind of equivalent being able to refer to particular releases. And that doesn't mean we can't have continuous improvement, but you can have frozen releases against which you download and do analyses. And then another thing, we had the general topics for discussion were around how we get sufficient terms, how we make annotation easy, how we make it easy to use the annotations once they're on the data set, and how we can work together to make atlases out of that. So that on that first point, there are many new cell type claims that come through these pipelines. We need sort of standard pipes that then take that into the cell ontology. And we have this provisional cell ontology as well, which gives us kind of a semi-automated way to get claims of cell types with links to data. And actually just on that point, we're hiring for ontology editors as well. So one good way that we can improve this workflow is to get some good biologists actually adding the terms. And yeah, I'll hand over to Kati for the other half the recommendations.
Thank you, David, and thanks to everybody who contributed to this session. It was wonderful to see everybody in person. Actually, if you click on that link in the lower right, you will also get to the slides. There are like 90 slides that were presented during that session on all kinds of topics, including how to use ontologies to improve cell annotation, but also cell segmentation. Ultimately, these two have to go together. And of course, whenever you improve the ontologies, then you will have to rerun, because now you have a different prior to what you're doing. And so how to do that version control, I think, is a really, really interesting part of it. And also how to minimize how often we clean data and interlink data is also very interesting. I wonder, just for my interest, how many of you are using knowledge graphs? I want to talk with you, please. We are losing out if we don't embrace this, because we want to interlink the different ontologies that exist to the atlas, of course also to our experimental data, but then also to other communities which have developed their own knowledge empires. And so yes, I would love to talk. So during this session, lots of good comments were made on absolutely retain original segmentations, but also annotations. We can always do 10 more, but ultimately maybe agree on the most relevant one for a specific task. It might very well be that you have different annotations and segmentations for specific tasks or user needs. I actually expect that, just like you have different world maps that serve different needs. So I think the atlas ultimately will have different views, if you wish, for different usages, but keep the original one. So that's now on the right-hand side under other recommendations. Also, oftentimes our algorithms now do provide confidence scores, and those should not just be shared with the portal owners, but also should be made visible to those which want to use this data. And even though some of, or not everybody might be able to create these scores, it would be great to at least know, yeah, that's a high confidence segmentation or annotation or rather low. Similarly, it's really interesting for us to figure out how to integrate the Uberon and FMA and cell ontologies and others to spatial data, which we now have more and more available at our fingertips. And there are some canonical data sources, 3D reference organs, 3D functional tissue units, 2D functional tissue unit references. We saw the anatomograms this morning presented by Irene. We had Melissa present 2D FDUs during our session. But how do you link those now to the Uberon ontology, for instance? And then I'm a great proponent for algorithm comparisons, so it was great to see that there are now many comparisons happening. We just opened another HubMap and HPA hacking the human body Kaggle competition. There are $60,000 prize money behind, so please come in and enjoy working with some of the best algorithm developers out there. And it brings in new expertise from other teams as well, not just this community here. Thank you all.
Thank you. Any questions for group 3?
Thanks a lot for the overview. I'm really curious, you mentioned in between, David, that one of the things that was discussed was should be reseeding annotation with kind of consensus integrated atlases. What was the result of that discussion, and how would you do that if you have kind of a separate hierarchical annotation and then putting that into an ontology? Will that always fit?
So maybe there's two questions there. One is the strategy for annotation and how we bring users into annotating these consensus atlases. And actually, yeah, and then the second one is the pipeline from that back into ontologies. So I guess with the brain data standards, we've got some experience of that already, where the annotation was all on consensus, integrated analysis from, I think, 11 or 12 different data sets all integrated, so multiple single cell from the same region, for example, certainly for the mouse. And there we were confident enough in the annotation, and because we're defining a lot of novel cell types, to make this pipeline into this provisional cell ontology. I don't really know the answer to the more general question of how this was more a question that came from Fabian. Lots of people are coming in with their original analysis, one data set, we annotate it on CAP, but then how do we bring users into the loop when we're actually building these really cool integrated atlases like the lung atlas that Fabian's been working on. So I don't know the clear answer to that, but I think that needs to feed into our decisions about what the workflows are that move data between these different platforms. So you could imagine moving that consensus back onto CAP and then opening it up for people to comment on and to improve on.
Thanks.
I think we should move on into the next group, group 4 on on modeling genetic diversity, and this group was led by Shyam Prabhakar and Patricia Severino, two members of the HCA Genetic Diversity Biological Network, which will also feature prominently in the next session.
Sean. All right. To motivate, I'm Shyam Prabhakar, Genome Institute of Singapore. And can you remove your mask? Is that okay? Good idea. Is this better? That's better. Yeah. Shyam Prabhakar, Genome Institute of Singapore, and I have with me Patricia Severino. We're representing the modeling genetic variation session. For some background, what are we trying to do here? As part of one of the key goals of the Human Cell Atlas, we want to model human diversity or healthy human diversity worldwide, and also make the atlas more equitable, more representative of all populations. And really for medical reasons, scientific reasons, I think there's a strong consensus that it is important to characterize human diversity comprehensively. And so that's motivation. The Genetic Diversity Network within the Human Cell Atlas was formed to address this challenge. And a big part of our task is actually defining what a genetic diversity atlas looks like. What does that even mean? And that did come up in the discussion. We decided to focus on the immune system as a case study for one very simple reason, that a lot of teams have been funded to profile diverse cohorts, to profile immune cells, specifically PBMCs, in diverse cohorts. So we said, let's take that as a starting point. The samples are easily available, lots of data available, and if we can come up with some paradigm for how to model diversity based on that, then we can extend it to other cell types, other tissue types. And also, we want to contribute to all the organ-specific atlases by helping them make those atlases diverse from the outset, rather than starting out with non-diverse atlases and later on layering on the diversity. And what do we mean by diversity? At least for the many axes of human diversity, the four we're primarily focusing on are ancestry/genetics, age, sex, geography. And there's more, but this is the starting point. Also want to say healthy atlas, as in everybody else's case, doesn't mean that they have no maladies or whatever. It just means not selected for any specific disease. And so Patricia can dive into all the takeaways.
Yeah, so we wanted to manage the group with some directed questions, but in the end, the discussions were very organic, so we just made a list of takeaways from the group discussion, right? So first, deliverables. We agreed on the effect of ancestry, genetics, age, sex, geography on cell abundance and cell states, gene expression, and it requires the integration of data analysis across all groups and projects. And a very important aspect that came up is that there is no specific funding for this integration, so we believe that this has to be discussed more deeply. Ancestry, genetics, ethnicity, culture, and race, as defined by social perception, were discussed a lot in this group, and they are distinct, and they are highly, very often confounded. Molecular differences between ancestry could actually reflect effects of socioeconomic status, lifestyles, differences between ethnicities, etc. And that's very hard to disentangle during the analysis. But self-reported categories can be useful, even though in the future they have to be associated with ancestry data generated by genotyping data, whole genome analysis, SNPs, and single-cell RNA-seq reads. Another question that always comes up is how many samples do we need in order to be able to get to this level of analysis? And the answer was we don't know. Like, nobody was really able to come up with something right now that can be used. So the effect of size is very important, and we cannot really solve this right now. when we talk about diversifying the atlas, that is something really challenging, and the discussion always came back to that, right? GWAS studies have not substantially diversified data in the past 10 years, so it's kind of difficult for us to think we're also going to be able to do that quick enough, but the idea is to really have that in mind and try to make changes that will lead to that. And we believe that this network could come up with recommendations to funders on how to identify funding gaps that need to be addressed in order to try to minimize this lack of diversity and make the atlas more equitable and inclusive.
All right, a few more takeaways. The very fundamental question, what does it mean to have a diverse reference atlas? Right? If you want to define an atlas of human cell types, if you do it in a non-diverse, very homogeneous cohort, versus if you do it based on data from all over the world, what is the difference in the output, right? If you integrate all of the data types using any of these integration methods, it'll put all those populations on top of each other. Fine, you define reference cell types. But then that reference cell type will have a different phenotype in each population. And then what do you say about that diversity, right? We have some answers to what Patricia said just now. Was perhaps one obvious thing is to say this is some kind of centroid in gene expression space of each cell type. And then you have these axes of ancestry, geography, age, sex, and you define how molecular properties change for that cell type along these axes as some kind of regression coefficients. But these are just initial ideas. I don't think anyone has really done this systematically on a large scale using single cell data. So open to ideas on what constitutes a diverse reference atlas. And how do we make this happen? So the community engagement came up, and that's a key thing. That's another reason existing omics efforts are not very diverse. It's not just that the research is done in geographically restricted areas. That's one reason. Another reason is even the diversity that's there in those places where the research is done is hard to access because some communities simply don't trust the researchers, right? Because of a history of exploitative studies that didn't really benefit them. And so what we've found in many cases is collaborating with researchers that have well-established cohorts, have good links to the community, have good community engagement. That helps us understand what the needs of the community are, what will bring them to the table, what makes them excited about participating in this kind of research. And on the same theme of leveraging existing cohort studies, right? These cohort studies, say UK Biobank, National Precision Medicine programs in various countries, they spend a lot of money, a lot of resources, a lot of time in generating whole genome sequence data and collecting massive databases of comprehensive metadata. And all of that metadata will be useful for us in understanding the determinants of human diversity or determinants of how our cells look different from other people's cells, right? So obviously we want to use all of that resource, all of that investment by adding. So the logical thing would be that we go to them, to these national precision medicine programs, we say we can add a single cell data set to some subset of your cohort. In return, please let's share your metadata and share your whole genome sequencing data. Then we can have a very rich data set, and that's a win-win. And we've seen that happen in many cases. Then down to the nitty-gritty of methodology, which we actually did not get into so much in this discussion, because we've had many other focus groups and discussion groups on methodology for this. Also, our methodology will align, we'll have to harmonize with all the organ-specific atlases. For example, we're working with the Immune Cell Atlas network with Chloe and others quite closely to say, okay, let's define a shared methodology for defining immune cell types. So it's not like we do it our way and you do it your way. Let's just do it one way. And then we can borrow their definitions of cell types and add on top of that, using that as a base, add on top of that knowledge of human variation. So that way there's no duplication of effort. Right? And again, for data integration, there's a lot of methodological discussions you could have about data integration, but perhaps we can say even for that, it's not directly necessarily a topic for the genetic diversity network. It's something that each organ system will develop, and we can exploit that further on. And yeah, so those are the main takeaways. It was very rich discussion and open for comments.
I think we have time for one question. We have one over here.
So it's, half a question, half a comment. So again, like a broken record, a lot depends on what you want to query. I mean, you brought both genetic diversity and I don't know, how does altitude influence cell types in the lung, which is one type of diversity, and another type of diversity is, being able to query like a GWAS, causal variant. And I've seen both discussions in this group, and I think they're trade-offs. So I think, as you're prioritizing samples, you have to really prioritize what type of queries do you want. We don't have endless resources, and this is a suggestion which I think is very important to think about, especially as people are building their roadmaps right now. In the past, we were talking a lot about the rare cell types and how do we ensure we sample a single individual deep enough to get the rare cell types, where I personally think all the interesting biology is happening. And I think also a lot of the variation that we're going to see that is functionally critical is in the rare cell types. So one can think of, and you said, define cell types, as we try and expand to more individual, maybe we should do more targeted sorts rather than, build every individual from scratch, oversampling all these very, very frequent states, which, we've had enough of them. So I really think that if we really want to get the diverse angle, we have to do some sort of sampling strategy rather than, unbiased sort for each individual, the diversity. So more a comment than a question, but.
I think Aviv has something to add to that.
I think I totally see the point, but there's two things to consider. First of all, it helps us also identify impact on cellular compositions, and you lose that with sorting. And there's beautiful QTLs to find for cellular compositions. Right, so that's one point, not to just do sorting. The second is that there are ways in tissues that have a large proportion of common cells and few rare cells to actually take approaches like overloading, but completely to the max, and basically by this way do label-free enrichment. The issue with sorting is that you need something to sort on, and that can be harder than it sounds. It also means cellular analysis, which is a serious limitation for a lot of cell types in tissues. There's a lot of ways to get rid of common things without actually doing sorting. So I just wanted to say that before people kind of go the traditional route of sorting.
I always look at what I meant in sorting is, throw some of your stuff into unbiased, at least 50%. But in the past, we were sorting each individual to crazy degrees to get these rare cell types. And so combine the unbiased and targeted for some of the really, targeted in any mechanism that works best. But I think that if we want to go to many individuals and we want to sample them at biologically meaningful depth, and we're not getting the entire world, the economy as our budget, we have to sort of make some trade-offs.
That's an excellent point, and more broadly, human diversity is such an impossibly infinite goal. And what we're starting out with is quite a modest initial step. Exactly. Exactly. So, as Dana said, some combination of unbiased and, say, selected or biased sampling would be ideal to cover the diversity. And we're really hoping, a call to all of you, please approach us and share your data sets with us. Both selected and unbiased, and so that we can do a cross-cutting analysis across cohorts. Because ultimately, the problem, so the GWAS community has recognized the need for diversity way before Human Cell Atlas existed. But their data sets are still not diverse. So how can we learn from that, right? So funding agencies need to get in to help us get there, and all of you need to help us with your data sets. I think that's a key priority.
Terrific, thank you. We have one more group then. Where are Ben and Ajay? So the final group is on not just data access, but user-friendly data access.
I'll start. Yeah, so we had a very interesting discussion. A lot of this felt like we were trying to fly the plane and build it at the same time. There's this constant tension between, well, what's the question and what's the answer? We kind of went back and forth. We discussed 4 questions broadly. Firstly, what is everybody already using? That was actually a pretty enlightening discussion. What are the different query types that we are all making day to day, and what is missing? What are the gaps? And that was also extremely interesting. And then we finished off talking a little bit about the challenges with multimodal data and how we might have to think about different queries differently for different kinds of data, but understand that people want to query, very general questions without necessarily thinking about the specifics of whether or not it's single-cell RNA-seq or proteomics or what have you. So we only have 1 slide to summarize, and I think you were going to do the first couple of points, so I'll let Ajay start.
So one of the themes that came about repeatedly was that there's a lot of techniques and a lot of methods, and people who are new and now even the old hands are getting confused about what to use for what purpose. And so there was a repeated request for better training and outreach for people. Essentially guidance on where to start to get answers for their specific questions. The other second point that came up was we all have, when we build various portals and user interfaces, everybody starts with common personas and use cases. And the different portals that Ben mentioned that we started off cataloging, that we should share what these user personas are and use cases are. And one specific item that came up is to build better and more facile collaborative annotation tools so that the community can do collaborative annotations of cells. So I'll hand the next points over to Ben.
Yeah, and just to put color on that first point, I think there was a general feeling about whether or not, for new people entering our field, whether we should be trying to think about HCA's position in creating training and outreach materials and where that stands. As we think about people onboarding through the DCP, what is their first experience with their data? And in terms of that user question that we were discussing, the fact that everybody is basically modeling the same, the same people and doing it multiple times, how do we kind of think about making sure that everybody has a kind of guidance for who they should be trying to serve when they build tools? In terms of expression-driven queries, we all see the need for people to be able to come in with a specific gene of interest, and there's obviously overlap with the technical discussion that was had earlier. There is, I think, actually good work that's going on now from various groups. There's a nice demo structure within the portal put out by CZI that allows people to kind of query by a starting gene and see where it's expressed, which cell types it's expressed in, and so forth. And I think that if we can kind of get agreement on the structure of the queries, the where and the how much is it expressed and in which tissues and so forth, then I think that is something that we can think about trying to add to our portal interfaces in our next funding period. And it feels like something that's relevant now that we have sufficient sets of data. We had a good discussion around computational frameworks. I think that we had an interesting comment was made that if you can't get a tool to work within the first 10 minutes of usage, then you give up. Right? That's a general truism of anybody in software engineering. And that we really do need to think more about actually the bioinformaticians trying to use the now hundreds of packages that exist in our space and making them actually not just easy to use for one particular query, but actually easy to get going on a specific machine. And so, yeah, there was broad agreement on that point. Finally, we discussed multimodal data a little bit. And while we all recognize that there is actually perhaps a lack of kind of truly integrated data sets, things are quickly building that
we do need to think about how we are going to essentially enable naive users to make or to ask questions of models that have been trained on data, and to actually build models from training data. So if somebody, there was somebody, I forget the name of the person in the group, but basically said, well, I want to bring together a set of different kinds of data types. I have no idea where to start. Well, it's really very challenging if they are, if they do have different kinds of data which have different kinds of error characteristics and so forth, how they kind of would bring that together and then train a model. And we really need to think through the sort of stages of, if you will, building an atlas, not just as a special kind of overall group, but as how individuals can build their own mini atlases of multimodal data and then ask questions of it and power their use cases and queries. And there was a million other things that we talked about. There's actually lots of good notes, but I think we will leave it there.
I think wonderful.
URL for the notes is
Yes, any questions for group 5? There's one in the back.
Hi there, Pablo Garcia from TCI. Thanks for sharing all of this. One of the takeaways that I got from the session early in the morning was that there's a lot of groups doing a lot of overlapping efforts in terms of data portal. And I was wondering if you guys touch on any sort of practical considerations that we may have as a community to both come together and share the knowledge, for instance, in terms of personas, but also to not create some sort of like fragmentation of the community because we have so many data portals. And there's some overlap in the data and there's also some overlap in the features, but there's obviously differentiated value for each of these different data portals. So I wonder if you guys discuss any sort of practical considerations in terms of that.
Do you want to go for that?
The only thing I can remember us discussing about that were the open frameworks, I mean the computational benchmarking that HCA has started. That was the only discussion we had that I can recall.
Yeah, I mean, I think that, so speaking from the DCP's point of view, so the coordination, we have a specific focus on producing and synthesizing together high quality human data sets and trying to create something with rich metadata and so forth, right? So we're trying to create data that has high value for that particular community. But we recognize that there are lots of other portals that are trying to serve different use cases and so forth. I think that, going back to that question about sort of training and outreach, it's definitely true that people need some guidance about what the best places are to start. Because when you talk to users, they'll often be, it's rather serendipitous. Oh, I started with such and such, and I find data from here and there, and get different answers from everybody. So I think, yes, I think it's a good thing for us to think through as a community. How do we attempt to deduplicate and ensure that what we're producing, what we're giving to people fits, if you will, right? That everything kind of is synthesized together. But I think that the core DCP certainly has an understanding of where it sits in the world.
Well, these clearly, I obviously was only in one of the sessions, but these were all clearly incredibly rich discussions that have been going on for quite some time and will continue to go on, but I think this is really an advance. I actually want to invite all of you to give yourselves a hand for contributing these ideas, and especially our moderators for the wonderful moderation job as well as the summaries that they just provided. So thank you. Okay, so we're now going to replicate the pattern we did this morning. We have a short break coming up, but before we get to that break, I'm going to tell you about the next group of the final session of the day, which is the last group of what we're calling working sessions. And then we'll go to the break, and then you'll go immediately to the working sessions, again, using the same 5 rooms that we used this morning. I was asked yesterday what the difference is between a breakout group and a working session. I also imagine that many of you who have been engaged with the biological networks are wondering what a working session is that isn't what you did 12 times over the last 6 months, because the biological networks are doing a huge amount of strategy work to develop their roadmaps for building an atlas, but also curating data sets, lists of data sets that could be candidates for Atlas 1.0, and also answering surveys, for example, on their computational resources that exist within the network, as well as where the gaps are in terms of their computational expertise. And so the title of this next working session is something like Bridging Data Production and Computational Integration. And I hope you'll see that we've really been building to this point throughout the entire meeting so far, because this is really where we want to take the work that the biological networks have already produced, the strategy work, the curation, and so forth, and really have a really deep conversation about how we're going to get from that work that's already been done and the strategy that's already been developed to integrated analysis and indeed cross-tissue analysis. And so that's really the point of this next group. So there are a few things that we're doing with this session that have been done ad hoc, but maybe not in such a concerted way before at an HCA meeting. One, you'll see that we've grouped networks together so that we're really having a conversation with 3 or 4 networks at the same time about these issues. These conversations will lead off with very brief, 5 minutes or less, presentations from one of the coordinators of each of the biological networks, really a progress update describing the roadmap, the data sets, and what their needs are going forward. These sessions will be moderated for the most part, by members of the HCA Analysis Working Group. And I sort of alluded to this previously, but each session will also have a representative of the Genetic Diversity Network included in the room. So you'll see that the Genetic Diversity Network is not on this list. That is because they are in all of the groups, and that way they can report back to the Genetic Diversity Network, and Sham has also offered to give a brief sort of response as part of the report back to these groups. So there will be, again, scribes in the session. There'll be staff to help with technical issues. I guess I would just say choose the network that you most closely associate with, figure out what the room is. Again, main hall, Heronsall, Perlatersall, Hartman down on the ground floor near the registration desk, and then the hotel room next door. And again, as before, when the bell rings, if you want to go to the hotel room, please convene here, and then you will be led over there. So thank you very much. We'll have about a 15-minute break, and then we'll restart about 4:10.